Hey y'all, so now we're doing the week 5 photosynthesis and respiration lab. There's only one lab this week, so hopefully that makes life a lot easier. Just make sure you're like on top of it and that you don't just say, oh, it's only half the work. It's going to be different, so be prepared, okay? And as always, read the manual, complete the lab, make sure everything's in the lab. You won't see this, you'll see this, which is the formatted document and the lab rubric, and then you're watching hopefully the playlist. So let me go to that rubric first. Okay, here's the rubric. So we got 3 exercise observations and then grammar and everything. So make sure you have complete sentences and all that. Let's go ahead and go over to our lab now that we know what to expect. So here's the week 5 lab. Title page, so your title, your name, the course number, the date. My name's optional. Oh, I guess I could open the key. I was being slacker. Oh, I hadn't done it yet. Okay, this is our onion one. Okay, I think of it as the onion. It's because of the next part. I'm just weird. La 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 la. Okay, let's see. Week 5 is... I know I could open it ahead of time, but I could also not bother doing videos, so don't get too complaining. Okay, so you have your table. You're going to say what color the suckers are, and then how far they moved, the RFs, the other student average for the RFs, and this is actually supposed to be 1. See, we're discovering things to move. Merge cells. There we go. And so that will just be the number of students. So that would be like, say you looked at 5 other students. There you go. Makes it nice. Okay. And then we're going to have our net photosynthesis rate, which should be more than the respiration rate. You add the suckers together, you divide by 10 and multiply by 60. Couldn't you just multiply by 6? You could, right? Whatever. Do it the way they want, but you could just multiply by 6. Silly. Because 60 divided by 10 is, sorry. Cheating. Don't want to confuse you. Just do what they say. It's fine. Either way works. Like crazy people. And then you have, was the respiration occurring during photosynthesis? Add back the O2 that was consumed by the plant respiration to obtain the total synthesis rate. So this is like, make sure you notice this. So respiration is always happening, yes. So then you have your rate versus the rate of the other, and then you'll put in how many others there were. And then down here, you're going to have that initial color, whatever the time was, the final color, the second time, and whatever that is in minutes. You can say 0, 60, like just do it that way, or you can say like 1 o'clock, 2 o'clock, whatever's easier for you. And then it's going to be your total time and then the average for everybody else, right? Yeah. So make sure these are all averages. So then the next is you construct a graph down here. So you want to do this as a stacked bar graph because it's going to look like your paper really well, and then you'll label a part. If you're not sure what a stacked bar graph looks like, Google it. You can just draw it by hand. Just if you draw stuff by hand, use a ruler, please, okay? Don't make it sloppy, make it neat. If you're labeling things, make it neat. Make everything really, really neat. It's annoying when it's all impossible to read. And it doesn't have to be huge, you know, size it so it fits correctly. It's big enough, not too big, that kind of thing. Identify the areas of the pigments that you observe on the filter paper. Are they located in the order that you expected? So was everything? If not, why? And as always, if not, why? Like, give it, hypothesize. Let's see. It makes me think of red onions. I mean, technically it's radicchio, but I'm clicking on things I shouldn't be, but it reminds me of red onions. Okay, so then B. Compare your strip with the radicchio, and does it have all the same pigments? You should be missing a couple. And then I gave you what the radicchio sucker looks like, right? Just so you have it, because it's nicely not in there. And then, if not, so what pigments are missing, and how much you have guessed this. So our radicchio shows you a picture of it. What color do you not see? So what pigment might it be missing? So then the initial hypothesis refuted, accepted. If you do not see separate bands of color, what might you conclude? So what may you still need to do, kind of thing. What might explain the separation of color? So why would they be separating? And then we have, how do your values compare to other people? If they're not the same, explain why. If they are the same, still explain how they could be different. So then we're on exercise 2, which is the conversion of light energy. So what molecules are produced when exposed to light? So exposed to light, does that mean that 2 processes are happening? Which organelles are responsible for that's what we should say, for these reactions? How about that? So there's 2 reactions. What are those reactions? Where are these molecules derived? Where are they coming from? Then, what was the purpose of the light? What does light have anything to do with anything? What molecule was produced by the Elodia, we're calling it Elodia, while it was wrapped in aluminum foil? So now we're down to 1 reaction. What reaction are we still doing? Which organelle was responsible for that reaction? And then we make up a table like 3. You can just make it look basically the same as 3. That's fine. Just add a table. Then name at least 3 experimental variables that could explain why your outcome might vary from other people. And then how could one determine how much oxygen might be produced in an hour? Like, how could we do this? So then it shows you, remember, you're always doing that inner diameter, right? So you need to find the volume, and it reminds you that P is this number. You're going to do your inside diameter of your tube. So the inside, not to the outside, just the inside, right? You're not going to include the actual tubing. And then you're going to square that, and you multiply that by the height of your tube. Okay? Squaring means multiplying by itself. So if it's 3, it'd be 3 times 3. And then you'll put here, and you'll show your work. Then to convert it to microlute, it's basically just converts over to show the conversion. And then so that would be per an hour, right? So now you want to know how long you need to produce 1 liter. So you know per an hour, so you need to multiply that by, let's see. You need to multiply that, how many microLs are in a liter, and then divide that by your rate, right? Because that would be, there's so many microliters, it goes through, so you take how many micros or liters are in this thing, divided by the rate you found out, and you should find out the time, because that cancels all our pretty little units, right? Yes, it does. I have to sit here and look at the math. And then, did the experiment support or refute your hypothesis? Voila. So chlorophyll, this one has a nice big chart. Chlorophyll gives plants their green colors. Research light and color. Hypothesize whether chlorophyll absorbs green light to any appreciable extent, and propose an experiment. So if you don't know, Google it. And then talk about an experiment that you should do to test this, right? You want to see if it absorbs green light. There is a current controversy as to whether the atmosphere is warming due to the burning of fossil fuels and the destruction of the rainforest, i.e. the greenhouse effect. How does the photosynthesis and respiration relate to this controversy? So if you're talking about the burning of fossil fuels and the destruction of the rainforest, how is that affecting, what is that adding to, affecting the environment or adding into the atmosphere? So how could photosynthesis and respiration help with this addition that we don't want? Why are the tropics particularly important to oxygen production? So is there a whole bunch of O2 produced there? Therefore, why would you support the actions to reduce the cutting of rainforests? Remember, you want to relate this back to our whole thing up here about why trees are good in terms of the greenhouse effect. And then while this exercise has addressed the oxygen production, what other important products result from photosynthesis? Besides the production of oxygen, why are animals dependent on plants? So why else do we need plants? other than just to breathe, because breathing's good, but we need them for other stuff too. Respiration and plants. So now we're switching over to that. See if I can talk about it 5 minutes. So why'd you have to rinse it? What gas did you add? How do you know? What's the, if you breathe, what comes out? Why did the color turn from green, yellow to yellow? Write the equation and indicate what colors on each side. So you check your lab manual, it's probably have an equation, and then which side of that equation is which color. What color change happened then? What gas was reproduced? What gas was removed? People are usually good at that. What process is occurring in the first trial without the aluminum foil? I'll say what processes. Make it obvious. I know I made it vague, right? This key definitely wants you to notice that it's more than one process, but the lab acts like you shouldn't know. It's like it wants to trick you, which doesn't seem very fair. And then what did you observe when you repeated it? Where'd it go? Blah, blah, blah. Yeah. Okay, when it's in tin foil now, what was the gas produced? What term could be applied to the second trial? Which process took longer? Why? And we're up to the discussion. How did the time change vary from aluminum foil to not aluminum foil? It should take longer with the aluminum foil. I will just tell you that. Okay, then why do both plants and animals respire? Why do we have to do that? Would life in either be possible without respiration? Assuming that we are using only this sort of chemistry. Because in extreme environments, you could come up with other options, and there are creatures that do. Organisms, let's say organisms. What was the initial hypothesis, refuted or supported? And then heating causes your indicator solution to change from yellow to blue very rapidly without the plant present. Why is this? So what is it doing to your solution? Reminds me of the buffer lab, right? Blah, blah, how did you control for this? So how did you keep the heat away from your solution? Then you have your data in another table. And then we do, were your results similar to the other students? What trends do you see? Hopefully the foil takes longer. Explain why the data may have differed from that of the other students. What variables could explain the differences? That's when it's nice if someone really did something weird, because hopefully they'll let you know. Or like they had a messed up plant, or just like a totally different plant. Sometimes it's fun. How would you be able to check to ensure that it was the plant that facilitated the color change and not some other factor? So could you take the plant out? Could you do something else? What could you do? So then there's other plants such as pine drops, Andromeda, ghost plants, blah, 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 that are not photosynthetic. So I might Google them to see how they make their food if you just don't know. It reminds me of my little air plants, but even them I have to fertilize once in a while. I just got a really cool one. There's a non sequitur. I like the air plants, they're fun. And then how do these plants still respire? Are they living? What did you say earlier in the lab? And then as always, lab summary. I can talk. Lab summary. You want to say, so all this taught you about photosynthesis and respiration. So 3 sentences about what you learned. Be positive, be upbeat, tell me things you actually learned. Summarize the experience. Be helpful. I'd probably do a sentence on photosynthesis, a sentence on respiration, maybe a sentence on that light thing, whether or not they absorb green light. I don't know if I would have realized that if I wasn't, a biologist. Okay, so how does that all sound? Hopefully it helps. It makes it all feel doable, possible. I'd love to review a draft before noon on the due day, so feel free to send me one.